Hello, this is Charmaine Espinosa, and I will be discussing recent results that we have generated from an experiment to determine effects of a novel phytase variant on nutrient digestibility in diets fed to young pigs. A traditional U.S. swine diet mainly consists of corn and soybean meal, and these feed ingredients contain phytate, which consists of 6-phosphate molecules bound to a myoinositol ring. Pig diets may also include feed ingredients such as canola meal and rice bran due to the availability of these feed ingredients in some areas. And if you take a look at the phytic acid profile of these ingredients, corn and soybean meal contain 0.5 to 1.7% phytate, and these concentrations are less compared with canola meal and rice bran. Therefore, if we include a significant portion of these feed ingredients in the diet, it is expected that the concentration of phytate will increase in the diets. With that in mind, we wanted to determine if supplementation of high doses of phytase is needed to reduce the negative impact of high concentration of phytate in the diets. Therefore, the use of microbial phytase in diets for pigs is a common standard practice to increase digestibility and availability of minerals from plant feed ingredients. Microbial phytase is pretty successful and well established in hydrolyzing those ester bonds between phosphorus and inositol ring in phytate. But in terms of amino acid digestibility, this has not been the case. Unlike in broiler experiments, where it was demonstrated that phytase increased amino acid digestibility, the effect of phytase on amino acid digestibility in pig diets is not consistent, and this may be attributed to differences in phytase source and inclusion rate of phytase in diets. With that in mind, a next-generation bacterial 6-phytase has been developed, and it is believed to increase phosphorus availability in diets for pigs. Indeed, in an experiment we conducted last year, we demonstrated that these novel phytase increased mineral digestibility and bone ash of pigs. We also compared this novel phytase with a different phytase source, and results from this study indicated that the novel phytase increased phosphorus digestibility more than the Butyroxella phytase. However, there is still limited data to demonstrate the efficacy of this phytase on nutrient digestibility. Therefore, an experiment was conducted to test the hypothesis that inclusion of increasing levels of the novel phytase in diets for growing pigs increases the apparent ileal digestibility of minerals, curd protein, and amino acids. We also wanted to test the hypothesis that the apparent whole tract digestibility of gross energy and minerals is increased upon inclusion of increasing levels of the novel phytase. In this experiment, we prepared 6 diets. First, a negative control diet that was formulated based on corn, soybean meal, and canola meal to contain 0.28% phytate-bound phosphorus in the diet. This negative control diet was also formulated to be deficient in total calcium by 0.15%, standardized total tract digestible phosphorus by 0.16%, and in standardized ileal digestible amino acids by approximately 0.02%. 5 additional diets were then formulated by adding 250, 500, 1,000, 2,000, or 4,000 phytase units per kilogram of the novel consensus bacterial 6-phytase, and therefore, we have 6 levels of phytase in this experiment. 18 pigs with an initial body weight of 17 kilograms were equipped with a T cannula in the distal ileum. Pigs were allotted to a 6x3 incomplete Latin square design with 6 diets and 3 11-day periods. There were 3 pigs per diet in each period, and therefore there were 9 replicate pigs per diet. The initial 5 days of each period were considered an adaptation period to the diet. For each period, fecal samples were collected on day 6, 7, 8, and 9, whereas ileal digesta were collected on days 10 and 11. These samples were then processed for nutrient analysis for us to be able to determine the apparent ileal digestibility of curd protein, amino acids, and minerals, and also the apparent whole tract digestibility of gross energy and minerals. Moving on with the results of this experiment, this slide shows the apparent ileal digestibility of calcium and phosphorus in experimental diets. Just to briefly set up the slide, here in the y-axis we have our response parameter, and in the x-axis we have our 6 dietary treatments representing the increasing levels of phytase. Blue bars are for calcium, whereas orange is for phosphorus. And what we can observe is that inclusion of increasing levels of microbial phytase quadratically increased the apparent ileal digestibility of calcium and phosphorus in diets. Formation of phytate complexes reduces the solubility and digestibility of these minerals, and therefore the observed increase in mineral digestibility indicates that the novel bacterial phytase was able to hydrolyze the calcium phytate complexes as well as some of the ester bonds between phosphorus and the inositol ring of phytate. We can also observe here a quadratic increase in the digestibility of curry protein with increasing levels of the novel phytase in experimental diets. Protein phytate complexes may occur due to electrostatic interactions between the negatively charged inositol phosphate isomers and positively charged groups of protein. Therefore, the increased concentration of phytate in diets due to inclusion of canola meal, which consequently increased phytate to protein ratio, likely increased the positive influence of phytates on protein digestibility. And if we dive deeper and take a closer look on amino acid digestibility, where lighter to darker bars represent the increasing level of phytase, we demonstrated that there is a quadratic increase in amino acid digestibility for lysine, methionine, threonine, and tryptophan, as inclusion rate of phytase increase in the diets. This is something we do not usually observe with all our phytase experiments, but with the current study, we use relatively younger pigs, and therefore it's possible that the impact of phytase on amino acid digestibility is greater in younger pigs than in older pigs, but further experiments are still needed to investigate if body weight of pigs influences how phytase affects digestibility of amino acids. These graphs shows the apparent whole tract digestibility of calcium and phosphorus in experimental diets, and here we can also observe a quadratic increase in the digestibility of these minerals, which also validates the results that we have generated for the apparent ileal digestibility of calcium and phosphorus. We have also determined the effect of the novel phytase on energy digestibility, and here we can observe that increasing levels of phytase linearly increase the apparent whole tract digestibility of gross energy. Phytate may negatively influence starch digestibility by inhibiting activity of digestive enzymes and by chelating calcium that is required for amylase activity. Therefore, we hypothesize that the observed increase in digestibility of gross energy upon phytase supplementation is likely due to increased degradation of phytate, which consequently increased starch and energy digestibility. In summary, we have demonstrated that the novel phytase increased the apparent ileal digestibility of amino acids and minerals. We have also demonstrated in this experiment that the novel phytase increased the apparent whole tract digestibility of gross energy and minerals. And these results are likely due to the ability of phytase to hydrolyze phosphate groups from phytate, reduce the anti-nutritional effect of phytate, and consequently increase energy and nutrient digestibility. Based on the results we generated from this experiment, in comparison with the inconsistent effect of phytase on amino acid digestibility, we likely need to revisit and take a closer look on some factors that may influence the effect of phytase on nutrient digestibility. It is possible that the impact of phytase on amino acid digestibility is dependent on composition and nutrient levels in our diets. Concentration of phytate and phytate may also play a huge role, and it's also possible that pig maturity can influence the effect of phytase on digestibility, and therefore, further research is needed to investigate if and how these factors influence phytase efficiency on amino acid digestibility. With that, we would like to acknowledge IFF for their valuable inputs and financial support. Thank you for your interest in this presentation. And if you would like to know more topics about pig nutrition, you can always visit our website at nutrition.ansai.illinois.edu.